Welcome to the ACNR podcast. Thanks very much for making the time for us. My personal interest is in how someone like yourself, very eminent in ophthalmology, started off and how the journey progressed. Maybe if we go back to when you were a medical student in Cambridge, is that right?
Yes. Well, first of all, thank you very much for asking me to record this for the journal and for the interest. So I would go back a little bit further because it was really an interest in science at school that was my main motivator. In fact, I decided when I was at primary school that I wanted to be a biologist because I'd read some books about animals and really enjoyed it. At the time, I also wanted to be a part-time tap dancer, but fortunately I gave up on that one. So I always had that. And then when I went to secondary school, I did all the sciences, and that's what I wanted to do. And the main reason I decided to apply to do medical sciences rather than natural sciences in Cambridge, because quite frankly, people told me that I would need to earn money because I'd come from a very well-off background. And quite literally, the teachers at school said you'd be better off doing medicine because you don't have any financial backing. And there was a bit of a tradition in British science, certainly in Cambridge, that you almost had to be independently wealthy. And a generation before us, really, many of the people were independently wealthy. So there was a little bit of that idea. So I didn't decide to go into medicine because, for the usual reasons, because I wanted to help people and heal people and so on. Although I could see that as something to look forward to, but it was really just because I wanted to do science, and people suggested to me that, I remember someone saying to me, you could be, even if you end up in pure science, you'll be paid more because you've got a medical degree. And he said to me, as the 15-year-old boy, he said, you could be sitting at the same bench doing the same research and you could be earning more than the scientist because you've got a medical degree. I don't want to sound as though I had a very pecuniary approach to it, but that seemed to be quite a practical thing. And even if I didn't end up practicing medicine, it would be useful to have a medical degree. However, when it came to it, I was really most interested in physiology. But unfortunately, physiology at the time was just beginning to fragment. Our lecturers a lot of them were still involved in whole animal work and this type of thing. And I did some projects of that type. Discovered that I really didn't like working with anaesthetized animals at all.
Just on that note, I do remember Professor Hickson, who was our physiology supervisor at Pembroke. I think he was supervising for nearly 30 to 40 years, and he was someone with a very unusual background of having a very good knowledge of physiology across species. In fact, based on what you've just said, I can understand how physiology has progressed and why they felt such a background was important.
No, that's right. But also physiology in Cambridge at the time, the mammalian side was dominated by the vets, in fact. So I was moving away from that, and I realized that actually physiology was all going to be done in biochemistry, and genetics wasn't really invented then in the way that it is now, but I saw that this type of work was going to end. But what I became interested in then was the psychology, because physiology in Cambridge was unusual because it was strongly connected with psychology. There was actually a corridor that went from one building to the other. They were that connected. They were next door to each other. And in vision science, there were the people doing psychophysics, which was all human studies. And this is what interested me, because I thought, well, I can use this in medicine. This is doing research with humans, gets away from the animal side of things without having to do wet lab work, having to work in a wet lab. And it was because I became interested in vision. At the time, there was Horace Barlow and Colin Blakemore and John Robson. There was between psychology and physiology, there was a big vision unit, Fergus Campbell. And that's what I wanted to do. So I did part 2 physiology. And then after I started training in neurology, I went back to physiology in Cambridge to do my MD, but did it all on human studies and psychophysics. That is really the route that took me from science, medicine, physiology, human physiology, and vision.
And specifically, the research that you did for your MD around vision in humans, could you tell me a little bit more about what it involved and what it was about?
Yes, well, optic neuritis was the topic. But the object was that I started studying that. Of course, at that stage, I'd already started my training in neurology, been through all of the general neurology training, but I'd always retained this interest in vision. And I started to do clinics at Moorfields at a very early stage.
As a registrar, or?
No, just entirely as a volunteer. There was no official way of doing it. But during my 3 years in Cambridge, I used to go down to Moorfields every Wednesday morning. The 5:30 bus from Drummer Street, and had breakfast somewhere near City Road, and did a clinic there with Ian MacDonald. It was neuro-ophthalmology clinic. It was what they called then the physician's clinic. There was no such thing as neuro-ophthalmology as such. It was called the physician's clinic. So because I realized I would need to know more about ophthalmology if I was going to be a neurologist with an interest in vision. But that's really how it all came together. And the actual research I was doing was all testing patients with residual damage after optic neuritis, looking at the residual visual deficit using visual evoked potentials, but also the pattern ERG, which was very new then. And in fact, we were the first to use it clinically, really, the pattern ERG, but also psychophysics.
So you completed most of your registrar training. You started gaining experience in the physician's clinic at Moorfields, but you also had the research that you were doing for your MD around neuritis.
Yes, from being an SHO in medicine, I went to a registrar job at Addenbrooke's in neurology, which I did for 2 years, but I had trouble getting back into Queen Square because I hadn't been a medical registrar. In those days, we all had to do at least a year as a medical registrar.
I see.
So I managed to get someone to give me a 6-month medical registrar job, Bury St Edmund. But basically, I'd then been in Cambridge for 5 years because I'd been 2 years as a clinical registrar and then another 3 years in research. So when I tried to get back to Queen Square, in those days, of course, there was no run-through training. Training was based on which jobs you got as you went along. And by then, they'd forgotten about me at Queen Square because I'd been away for so long. But anyway, after I had to go and do this 6 months as a medical registrar, and then I got on the house at Queen Square. And then, of course, I'd been in SHO there, but they'd forgotten me. And then I started to work with Ian MacDonald then, you see, because, of course, he was very interested in optic neuritis. What made me continue in neurology was working with Ralph Ross Russell, who I knew as an undergraduate, because he was on at Thomas's as well as Queen Square, and I was a medical student at Thomas's. And I just thought his teaching, although it was rather understated, shall we say, was incredible. Incredibly precise way of thinking out the problems and so on. And I knew he worked at Moorfields. So I realized then that you could do neurology and vision. And then my SHO job at Queen Square was with Chris Earle, who's the consultant. Roger Bannister and Roman Cosin were the other two. And he also worked at Moorfields. So I saw all these really fascinating eye cases coming in as an SHO. So as then at that early stage, I could do neurology because ophthalmology then, you see, was a very, very surgical
specialty. Yeah.
In fact, you had to do general surgery to get into ophthalmology. So again, no run-through training. I would have had to be an SHO in general surgery and do FRCS. Now, of course, ophthalmology training is quite separate from the surgical training. So all of those different things converged to keep me in neurology and keep me in vision science and keep me doing this, not doing animal research. I also did animal research when I was a medical student at St. Thomas's because there was a very good vision lab there run by Hisako Ikeda,
Okay.
And during my 3 years as a medical student, I spent all my spare time working in her lab.
I see.
But that again was animal work. It was anesthetized cats.
Mm-hmm.
And she had a model of strabismus where she used to remove the lateral rectus muscle from one eye of the kitten and then allow it to develop. And then the animal was sacrificed and the recordings done. And what I did was the histology. And in fact, my first and last paper in Journal of Physiology was with her when I was a medical student. So one way and another, I mean, a lot of it just by looking at opportunities rather than anything that was offered to me or anything that was available through training, I managed to keep going in vision science while I was doing all the right jobs to become a neurologist.
You were using all the spare time you had outside of a full-time training post. I mean, that's dedication.
Medical student. I'm talking about spare time as a medical, but that was also
Well,
true.
you seem
No,
to have continued to do that during your neurology registrar training.
No, that's true. I did a lot of research when I was a registrar as well at Anbrooks and later on at Queen Square, mostly because, and it was all entirely patient-led, and a lot of that was nothing to do with vision either. I just went wherever the patients took me,
And what happened after you'd completed your training, your research degree? How did your career as a consultant start? Where were you?
Well, I really wanted to go to America.
Oh, right. Okay.
Really, really wanted to go to America. That is something that I'd been planning for years. And I realized when I was senior registrar at Queen Square, then you do have quite a lot of time. You're quite busy when we all did a year out and then a year. I was at the Middlesex and then a year at Queen Square. And actually you were never very busy because the registrars tended to do most of the ward work. In fact, a lot of the SRs were never seen on the ward. So you had your own outpatients and you ran your outpatients as a sort of like a junior consultant. So I had quite a lot of time then as well, but I was really, really keen. I felt that I was secure enough because there wasn't really anyone else around who'd done the ophthalmology. I mean, there were a lot of people who were interested in vision, but there wasn't really anyone else who'd done the ophthalmology. So I felt I was quite secure, and I knew that Chris Earle was retiring and Ross Russell retired a little later, but Chris Earle was retiring and I did his locum. I did his consultant locum at Moorfields, at the old Holborn branch. So I felt fairly secure. So that was my chance. So I applied. I'd been supported by the Wellcome when I did my 3 years in Cambridge. And then for various reasons, I was out of favour with the Wellcome. And so then I went and they turned down my application, but I took it to the MRC and I had no trouble with them.
Mm-hmm.
So I had a year in San Francisco in a research institute there called the Smith-Kettlewell Institute. And that was a wonderful year. We arrived with a 6-week-old, a 3-year-old and a 4-year-old. So
you had a family by then.
Yes. But I don't know, that age, when in your 30s, you just sort of take it in your stride, don't you? And Marilyn didn't mind having a year out. She was in the practice that she wanted to leave anyway. So she resigned from the practice and spent over a year. Actually, we spent nearly a year and a half in San Francisco. By this time, Queen Square was saying, we want we wanted to start this job. And, at that stage, they advertised I was the only applicant for the job.
Yes.
It was me or nobody at that stage. But I found being in the States a real eye-opener in terms of the difference in research, because this was a private research institute. It wasn't a university. In fact, you couldn't do a PhD there. You had to be in Berkeley or somewhere, registered for PhD and do your research there. And all the people who worked there knew that if the didn't get their grants, they didn't have a job. It was just so different from working in the university department in Cambridge, where actually everyone was on tenure of one sort or another. And it was only the people who didn't have tenure who were really doing all the work, and professors were a lot of them sitting back on their laurels, and certainly the technicians had no particular motivation to help you. Whereas it was a real can-do situation there. They all really wanted to help, right down to the secretaries and the receptionists and the graphics department and so on. They were so helpful, and that was great. And I worked with patients there as well.
What sort of research did you do there at the institute?
Well, I wanted to start working in the brain rather than just in the optic nerve. So, and I'd been looking at some aspects of motion perception, optic nerve disease, and wanted to expand that. So I had to take a chance that I would find some patients who had deficits in motion perception, because actually they're very, very rare. And the reason they're rare is because it's very unusual to get damage to the lateral occipital lobe where the motion area is without getting a hemianopia when you've got nothing to test.
Yeah.
So there were just a couple of patients in 100 years who'd had bilateral lateral occipital lesions and had lost motion perception completely. This was fantastically rare. I mean, I had no hopes of it, but I just had the idea that if we looked hard enough, we could find people who didn't have a hemianopia and had a motion deficit. And fortunately, I found 3 of them, which was enough, and did hours of testing with those patients, absolutely hours. They were wonderful. And it was such a great atmosphere there. So I didn't regret that at all. And then when I came back, I was a consultant. That was it.
So you were in a full-on clinical area since then? Or did you still continue to have a thread of research going on in the background?
Oh, yes, definitely. But it's very different. Once you start to get PhD students, it's very, very different from up until then. I've just been doing everything myself. And then what you find that you're having to do is decide what the student is good at, what the student is going to do best at, and allow them to go with that. And so in a way, you don't have such freedom with the research. And we did lots of things that were never published, which I always regretted, because some students are just not good at writing things up. They'll do everything else. And then there was other work coming in. But the other things that happened were, again, quite by chance. When I was an SHO at Queen Square, Peter Gautier-Smith, who died a couple of years ago, and he must have retired about 30 years ago, and he had collected a family with an amyloid angiopathy of the brain. They had we knew of one patient who was very ill in that anyway. So he just gave me this project. I started working on that, and it took 20 years. It was 10 years before the patient died, and another 10 years. And I had to work with a group in New York because Blas Frangione's group had basically cracked extracting the amyloid protein. And as you know, amyloid is a quaternary structure of protein, but there's many different proteins can form it. So you have to get the amino acid sequence of the amyloid protein. And the whole point about amyloid is it's insoluble. It's very, very difficult to get it out of the brain. And they had described the amyloid. They weren't the first in Alzheimer, but the Dutch amyloid angiopathy and the Icelandic. They'd all gone to this lab. And so I contacted them, but it was 10 years before the patient actually passed away. And we were able to collect the brain half frozen and half fixed. They did a fantastic job, but they didn't get a complete amino acid sequence. So we were actually spent 10 years looking at the Human Genome Project. Actually, there was a message went round on the consultants chat saying, it's amazing. The patient came in and then someone came in and consented them and took the blood, and they were already having a whole genome sequence being done, having been seen in the clinic that day. Well, we didn't have a complete sequence, and we had to wait. Every week we looked at the Human Genome Project to see if we could find a match. It was 10 years. So from starting the project, 10 years till the patient died, and then another 10 years before we got the sequence of the protein. I mean, of course, if it had been today, it could have been done
Everything's much
overnight.
faster.
Yes, but it felt like more of achievement. And so I also had this interest in amyloid angiopathy, which didn't actually have anything to do with vision. And the exciting little chance thing that happened there, I used to teach every year in Denmark, in Aarhus. The format there was quite interesting because we weren't told anything about the patients. And then a pair of the students would take a history and examine the patient and then come in and present the history and the signs to us. So it was sort of the opposite way around from Gowers, where the consultant knows what the diagnosis is and the registrars don't. Anyway, so we were expected to get the diagnosis right. So I've been doing that for a few years. And then one year they showed me this patient who was exactly like our British, what we by then had called British familial dementia. The Danish patient was exactly the same, except that very early on, they had had cataract and angiopathy of the retina and deafness. And I said, oh, this is so like our familial case, but also has these differences. It's a very small pedigree compared to ours, but it was the same situation. We had this older member of the family, the dad, who was very ill, and that took another 10 years. And quite literally, I was at a neurology meeting, I think it was in Rhodes or somewhere like that, and I was talking to Brandgaard, who looked after this patient, and I was saying, how's Leif Kroner, oh, he's still hanging on, we're still waiting. And as we were talking in this bar, he died that night.
Yeah.
I mean, sadly. And so we got the frozen brain. By then, we already had the other sequence. So it only took a couple of weeks then to find that was a different mutation in the same gene.
I see.
That's still going on. In fact, the paper's just come out on familial Chinese dementia.
Oh, right. Okay.
There's other, because this was the first dementia on chromosome 13. And there's also this same gene is also the cause of a retinal disorder, which has been reported in France. So, I mean, that's been a little thing on the back burner for me now for 40 years. And ever since I was an SHO at Queen Square, it's been another fascinating thing. But there again, I wasn't able to do so much of the work myself. And in fact, Simon Mead, he was about to start working with John Collins on the prions. And for some reason, John couldn't take him on. So he spent 6 months working with me and our family, because we then had to find out whether this mutation was the same, to do the LOD score. We had to look at the extended family. I was in contact with over 100 descendants of the patient, but they were in New Zealand, Australia, Canada, all over, South Africa. So Simon Mead went to all these, took samples and from all these patients all over. He didn't go to Canada, but of course, everywhere he went, he climbed the nearest peak. So we were able to get enough samples from enough of the pedigree to show that it was the causative gene. And as I say, that story is still going on. So a lot of these just happen by chance. Gautier Smith hands me this family tree. And I'm teaching in Aarhus and see a case and, bang, it's a whole story.
That leads me on to my next question. What would you say has been, do you think, in the last decade or so, a sort of a landmark discovery in diagnostics or treatment in the field of neurology and neuro-ophthalmology? Would you say it's more the progress made in genetics?
Well, not for me, because it's come late. It's come late in my career. As I say, genetics was moved in geological time when I was involved in it. And there were so many of these tantalizing families and disorders around. I mean, the different types of inherited optic neuropathy and retinal disorders, and there were clinical classifications of all of these things. And most of the people who were working in that field, it was more like stamp collecting. You collected your pedigrees. So it wasn't really so appealing then.
before I forget, I have to say, although I haven't had the chance to train with you since you retired the day before I started my neuro-ophthalmological rotation, I did have a look at your library in the little office in the eye clinic at the National Hospital, and did find an ophthalmology stamp album.
Oh, yes. That's right. Yes.
Sorry to interrupt, but it was a book that I had to pull
I
up.
can see how you've made the connection. Yes, I think in the early days, a lot of physicians were botanists, and in fact, actually my favorite subject at school was botany. Yeah, but I thought because of my name, I didn't think I could really.
Yeah.
You were Professor Plant, the botanist. It'd be like something out of Happy Families, isn't it? So in fact, I didn't take the A level. Even I did the A level course, but to get into Cambridge, it was more important to get 3 good than 4 A level. So I didn't take the A level. But I've always had an interest, and I still take it. I've got a notebook of plants that goes back 40 years that I've collected, so I can go back and look over. So yeah, I don't collect stamps though. But at that time, that period of genetic disorders, it was a lot like stamp collecting. And in fact, the person who was most involved in collecting family trees at Moorfields, Barry J, he actually had a stamp collection which was internationally known.
Oh, wow. Goodness. Okay.
Yes. And in fact, there was a famous occasion when, because, the Queen has a wonderful, wonderful collection of stamps, and there was something missing. And the palace actually contacted him to loan this stamp for some suspicion. So I think that was the mindset of a lot of people who were very successful in that period in medicine. But I think that if the advances we've seen now in genetics had happened 30 years ago, then obviously I would have been involved in that more than anything else. I mean, it's been amazing. But I think from the clinical point of view, because there is another argument about we're only just really seeing how the advances in genetics are leading to treatment. That's happening now. That is happening now in a big way. And that's clearly the way forward. There was no of that happening. And I think for the practice of neuro-ophthalmology, biggest advance really was in imaging.
In what respect? Do you mean MRI imaging or new tools such as ACT?
MRI and now OCT. I mean, I think OCT is just as important as the invention of the ophthalmoscope, which was, coming up to 200 years ago. Well, certainly 150 years ago. And that was a revolution. And it's interesting that many of the practicing ophthalmologists around were not impressed by the ophthalmoscope. And Helmholtz, who was one of the inventors, he said that he couldn't get ophthalmologists interested in it because they said they could diagnose all known diseases of the eye without the aid of his instrument, which of course was perfectly true. What they couldn't diagnose was all the unknown diseases.
Of course.
And yeah. I think it's the same with OCT. It really has moved things on enormously. But that came late for me. And more so than electrophysiology, although that's still in ERG, that's still advancing now. But I think in terms of what was most important for me in the clinic was MRI. And I did 3 hours a week programmed sessions in radiology. I did an hour at Moorfields, an hour at Queen Square, and an hour at St. Thomas's just with radiologists. And I found that what was so important about that was us being able to tell the radiologists what our clinical needs were, and them being able to tell us what they could do with MRI, which was, of course, always advancing. And I worked in Ian McDonald's group, which was to do with MS. In terms of how useful it is in the clinic, there's many more important things than MRI in MS, really. So I think the everyday practice for my decades in neuro-ophthalmology, I think MRI which of course was very new. I mean, when I was a registrar, it was very new. In fact, when I was a houseman at St. Thomas's was when the first CT scan was, the first one, as you know, was at Atkinson Morley. And then while I was a houseman at St. Thomas's, they put one in Queen Square. So, and because we had both Reggie Kelly and Ralph Ross Russell were both on the staff of Queen Square, so we started to set, but there was a huge wait, of course.
Okay.
And the CT scans, the images were on Polaroid film.
Oh, wow. Goodness. Okay.
The image was actually a Polaroid photograph of the monitor. There was no X-ray film.
And as cross-sections of the brain on each square.
They just cycled through the cuts, which were very thick compared to nowadays and took a lot longer to do. And then as each image came up on the screen, they took a Polaroid film of it. So that was the only permanent record of the investigation. It was very different then. But CT was a revolution. And then MRI even more so, really.
Yeah. And the more, I suppose, investigations, imaging tools are becoming more precise and sensitive as time passes. How do you think that might affect the role of a physician or a clinician in clinic?
Well, people are always saying that this investigation or that investigation makes the clinician redundant. I can remember when I was a neurology registrar at Addenbrooke's, one of the neurosurgery senior trainees said to me, "We don't need neurologists anymore. We've got myelograms."
They still say that in Addenbrooke's.
We've got myelograms. What he meant by that, they didn't need this examination, the type of examination that used to be done to localize the level of lesions in the spinal cord. And then the surgeon would, the picture of Gordon Holmes going up to theatre and putting his finger on the skull, and that's whether that's apocryphal or not, I don't know. But that was it. The surgeon couldn't do anything until the neurologist told him where the tumor was, you see. So he was thinking that that's all gone now with myelography. I mean, CT was not even being used so much at that time. But what I find happens is that it just takes you to another level of clinical expertise. I mean, a good example would be a condition like pseudotumor cerebri.
Mm-hmm.
When I look back to how we managed what we called BIH back in those days, the way we managed that, it was a very homogeneous condition to us, normal brain imaging and a lumbar puncture with a high opening pressure and slap Diamox and tell them to lose weight and so on. But we now know from imaging that actually it's a very complex situation where we didn't know why the pressure was high. We still don't, but we do now know how this feedback mechanism where the pressure is actually increasing the venous pressure. The dural sinus venous pressure is being increased indirectly, therefore causing even higher pressure, and how the optic nerve is affected and so on. And whereas I would say the clinician in the 70s and 80s was making it the same diagnosis in what we would now see as a range of conditions, where is the priority the obstructive sleep apnea, is the priority sinus stenting, is the priority optic nerve sheath fenestration, could there be a small dural fistula there? I think that it actually takes it to another level of complexity where your clinical expertise comes in again. But provided you work with the interventionalists and you work with the diagnosticians in genetics and in imaging, so that you learn from them and they learn from you. But I think every advance, just certain things become easier, but it opens up whole new depth of study and knowledge. So now I think we still need clinicians. Definitely.
And if you had to give advice to trainees like myself or new consultants as they embark on their career as a neurologist or a neuro-ophthalmologist? What would that be?
I think that if you're not really enjoying what you're doing totally, then I think that there's something seriously wrong. If there's any aspect of your work that, doesn't give a bounce in your step in the morning, knowing that you're going in to do it, I think there's something wrong there, because it is the most fascinating way of life, really. I wouldn't swap it for anything. Of course, I would probably, if I could have another life, I'd rather just do science and not have to deal with medicine.
Mm-hmm.
There's no doubt that as a clinician, with many exceptions, there are many really, really top-class scientists who've also been clinicians, but for lesser folk like me, the clinical side does take up a lot of time. But on the other hand, if I'd just been working on the neuromuscular junction or something, I'd have been, as a physiologist, I'd have been stuck with that. Whereas medicine throws at you googlies, throws at things at you that you didn't expect or other people have ignored, and take you off in other directions. But I think to be a really top scientist in terms of achievement, you have to be a bit more focused than that. And that's quite hard if you really enjoy medicine. But that's my main advice. So, and I think if there is any aspect of your job that you're not enjoying, then you should be changing it in some way, because there's something wrong there.
It is harder to remain as a general neurologist, I feel, in my time, with a research interest, which almost shapes a subspecialty interest. And, you're automatically seen as the MSologist, only doing MS or neuroinflammatory clinics, or maybe even now that the spectrum of MS disorders such as MOG, etc., as the spectrum increases, maybe things will become more and more super specialized. But yeah, I do like general neurology. I think it's going to be a tricky balance to find an environment where the subspecialty interest as well as the interest in general neurology will cater
Well, the
for.
model at Queen Square previously was that was expected to have some sort of research interest, but everybody also worked at a general hospital, you see.
I see, yeah. I suppose that would be the district general hospital contract that a number of consultants will have, and the tertiary.
Yeah, that's right. So I think if we're still going to have neurologists going out to EGHs, and of course for some of them it meant teaching hospital like St Thomas's, but still you would see real general neurology there. I mean, the teaching hospitals still act as major general hospitals. So I think that was the model, whereas now I think it doesn't quite work as well, and you find that most of the people who are in the institute now just want to be in the institute doing, and they would argue that you can't really do all that stuff and really be focused enough on your research to really achieve. Remember that there were very few professors in neurology in the past. And when I started in neurology, there were only 120 consultant neurologists in the whole country, the whole UK, 120 neurologists for 60 million people. So they were all busy, they all had their DGHs and they did neurology. No, I think it's clearly not going to happen anymore. I mean, someone like John Morgan Hughes, who was a world leader in muscle disease, and his big thing was you have to get a bit of tissue. You have to have tissue from your patients. That's why he worked muscle and not brain, because it was easier to get tissue. He said it was only an accident that muscle disease got into neurology anyway, but he was a real world leader. But he went to Bedford. His DGH was Bedford, and he kept us well supplied with subarachnoids and all the general stuff, and he loved doing that. He said you shouldn't give that up, but it's not happening that way anymore. It's people have got one disease or one group of diseases, and they just want to spend all their time in the institute.
Yeah. And how do you spend your time now, Dr. Plant, now that you're retired?
I seem to be quite busy. I have tried to do more music. I started having music lessons again after 40 years and joined a band. So I've been doing a bit more seriously music, and I still have PhD students. And some of this is just accidental because of COVID, because one of them ended up stuck in Brazil and another one ended up stuck in Australia. So their work has dragged on a bit longer than it should have done. Well, a bit longer, 18 months longer, really. But also a project that I started at Smallfields in optic neuritis some years ago has now been taken on by another PhD student. And I have a PhD student in Belgium. I'm supervising a PhD there. So that keeps me going. It's all on Zoom now. And then I like to still listen to the Gowers, and I like to listen to the medical eye unit at St. Thomas's every week. So I still enjoy those.
I hear you're due to present at Gowers soon.
Yes. Well, they've very kindly carried on letting me do a session every term. But now, of course, they're not my patients, but I enjoy. Yes, it's generally quite busy.
And spending more time with grandparents, more time to do podcasts like this one, which is great. Well, I thoroughly enjoyed the last 40 minutes or so, Dr. Plant. People speak very highly of you, and you are a true icon in neuro-ophthalmology. And it's amazing to see someone who is still engaged with education, research, etc., even after retirement. It really reflects the passion you have for your career and the science behind it. So thank you very much. Brilliant.
Thanks for listening. Don't forget to follow us on Twitter at ACNR Journal. Sign up for our email newsletters through the website acnr.co.uk, and you can also follow us on LinkedIn.